feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the National University of Singapore Yonglou Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us tonight. It's a little early for me. I'm back in the US in Texas, so I'll try to be coherent throughout the talk. And we have Dr. Kevin Perez with us tonight, and we'll be introducing him in a minute. I want to remind you to ask questions. Anything that comes to your mind, we'll try to collate them and get them to Kevin for some exciting answers. Before we go to that, though, I want to introduce Subha Subramanian, a research associate at the National University of Singapore, and she'll be talking about her own study unraveling the role of rapamycin in aging skeletal muscle, the impacts of intramyocellular lipids.
Hello, thank you for the introduction. As mentioned, I am Shubha, working in Prof Brian Kennedy's lab as research associate. So today I'll be talking about the role of rapamycin in aging skeletal muscle and its impacts on the intramyocellular lipids, which is termed as IMCL. So, as the image shows how aging affects the skeletal muscle with increased mTORC1 activity as individual age. This leads to increase in the mitochondrial oxidative stress, protein degradation, muscle atrophy, which contributes to sarcopenia. As we age, there is an unhealthy accumulation of lipids in skeletal muscle, which negatively influences both muscle function and metabolism. So the young muscle fiber is characterized by a minimal presence of intramuscular adipocytes, which are normal and serve as a source of energy. As we grow older, this leads to lipotoxicity, a condition where excess fat interferes with cellular function. In aged muscle, excessive fat accumulation is seen both outside and inside the muscle fibers. This disrupts normal muscle metabolism and contributes to insulin resistance and anabolic resistance. The study investigates IMCL metabolism and muscle mass with rapamycin in aging rodents. Male visitor rats around 12 months old on a CHO diet were split into two groups, with group 1 receiving the vehicle and group 2 being administered rapamycin. Coming to the result, in rapamycin-treated mice, the body weight decreased due to mTORC1 inhibition, indicating increased lipid utilization and reduced fat mass. Mice subjected to rapamycin administration exhibit diminished triglyceride concentration, thereby suggesting improved lipid catabolism and decreased levels of circulating lipids. The TA muscle is utilized for all these analyses. IMCL levels significantly decreased after 2 weeks of rapamycin treatment from the baseline. The levels of IMCL showed a considerable decline after 2 weeks course of rapamycin administration in rats relative to the control group that received the vehicle. These results strongly support the hypothesis that rapamycin improves muscle metabolic health in aging by reducing excess lipid accumulation. Coming to the molecular analysis part, MyoD and GLUT4 markers is increased, indicating improved muscle regeneration and glucose uptake. Myostatin and atrogen 1 markers are considered as a muscle wasting or notably reduced. P70XSK, a downstream effect of mTORC1, is upregulated potentially due to regulatory feedback. The rapamycin group showed an larger muscle fibers than the vehicle group in H&E stain sections. The histological results support our molecular and the metabolic data suggesting rapamycin's role in preserving muscle health during aging. The focus here is on the results of the experimental investigations, which we have already talked about. The key takeaways from the examined data is rapamycin's inhibition of mTORC1 could potentially address the accumulation of IMCL in aging skeletal muscle, which help to improve the muscle metabolism, suggesting it could be a promising strategy for enhancing the muscle health and longevity in older adults. Future research will focus on investigating rapamycin's long-term effects on aging skeletal muscle metabolism, safety, and dose optimization. So we'll be using an aged myotubes to investigate the synergistic effects of rapamycin with compound B and compound C on muscle vitality and metabolic processes. That's the end of my presentation, and thank you all for your attention.
Thank you, Suba. So I want to introduce Kevin Perez, who's a junior group leader at Charité Berlin. Kevin holds a PhD in computational biology from Charité University and has worked with leading experts in aging research at the Buck Institute and the University of Lausanne. He co-founded the Swiss longevity biotech startup Epiterna, where he led research efforts. And in 2025, he joined Charité Universitätsmedizin Berlin as a junior group leader, bringing deep expertise in aging and computational biology. The topic of his talk tonight is going to be lifespan extending drugs in model organisms and humans, something of interest to all of us. Thanks for joining us, Kevin.
Thank you, Brian, and thank you to everyone attending the talk online. I'm just going to share my screen. Okay, so today I'm going to talk about lifespan extending drugs in multiple model organisms, but also in humans. I'm going to present the platform that we've built using multiple different model organisms, including worms, flies, fish, and mice, to try to discover those drugs that could potentially extend lifespan. And I'm also going to talk about some work using biobanks to find drugs for humans, because I think it's interesting to do research with model organisms, but it's even better if it can also relate to humans. So, as you all know, aging is something we, I think, all want to find a cure or a solution for. We have a rapidly aging population, increased incidence of age-related diseases, and all of that poses a significant burden on society and healthcare systems. So the type of drugs that we want to identify, they could have three different purposes. They should prevent or delay the age-related functional decline. So as we age, we lose function, we lose autonomy, and we want to prevent or delay this as much as possible. We also want to prevent the occurrence of age-related diseases. And the last one, which is in a way also the most important, is we want to delay all-cause mortality, which is really the final, in a way, the final endpoint of aging. So as I said, we have established a screening platform for lifespan extending drugs to be able to find those drugs. We use a lot of different model organisms. We work with worms, C. elegans, Drosophila, which are flies. Killifish, it's a different type of fish that most labs work with. Most labs work with zebrafish, and we work with killifish because it has a particularly short lifespan compared to zebrafish. And lastly, in mice, which is, I think, what everyone uses for, most people use for mammalian studies. So right away, these models have very big differences. The most notable one is their lifespan, which is what is interesting us. So some of them are relatively short-lived. The worms live about 3 weeks. Flies can live 1 month and a half. Those fish live about 4 months, and then mice live 3 years. So that's much longer. And these models come with different sizes, different complexity, and with that also comes different costs and a different throughput, meaning that it's much easier to test a lot of drugs in worms than it is in mice. And it's much more expensive to test drugs in mice than in worms. And why did we build this platform? Because we have a hypothesis, which is that we can identify conserved anti-aging pathways between those different model organisms. So you could find some drugs that work in worms, then work in flies, then in fish and in mice. And those more conserved drugs could later work in human. And you can also use those small model organism as a pre-screening step before testing drugs in fish or in mice. So the first model I'm going to talk about is C. elegans. It's one of the most used model in aging research, I would say. So it's a nematode. It's quite small, and you can do very large-scale experiments with this. There are a lot of groups that are already be testing drugs to extend lifespan in C. elegans. There's a database called DrugAge, which is compiling all the drugs that have been testing for lifespan extension in multiple model organisms. And in this database, there are already 500 drugs that have been tested in the literature for extending lifespan of worms. And then there's a pretty significant reporting bias because almost 70% of those drugs have a positive effect on to increase lifespan. And I'm just giving you here 3 examples of those drugs that are, I think, quite famous: resveratrol, rapamycin, and metformin. But what are the issues with testing drugs in worms? I mean, in general, I would say that there is a lack of reproducibility. So if you ask different labs which drugs really extend lifespan, which don't, you will have many different answers from different labs, which I think is in general not a good sign. And also there are a lot of confounding effects. And one particular effect that I want to talk more about today is the interaction between the drug and the bacteria. So the worms, they are fed to maintain them. You feed them with a type of E. coli called OP50. And when you give them also a drug try to extend their lifespan, this drug is also in contact with the bacteria that is being fed to the worm. And it's difficult to know if when the drug is extending lifespan, does it act directly on the worm or does it act indirectly through the bacteria then on the worm? So these are the types of effects that we try to understand a bit better. So in this part of the talk, what we did is we curated 16 of the most reported lifespan extending drugs in worms. We curated them from DrugAge, which I mentioned before, but also in general from a review of the literature. And we tested their effect on lifespan of the worms with 2 different types of food. So the live normal OP50 bacteria and then a UV-killed dead version of the bacteria. And we asked, does the effect on lifespan depend on the type of bacteria being fed to the worm, which would indicate that maybe this effect is indirect and not direct. So these are all the 16 drugs that we tested. So very quickly going through them, you see that there are 3 mTOR PI3 kinase inhibitors, GSK, LY, and rapamycin. And obviously this pathway is very important for aging. there is metformin, there's 3 different antibiotics, and there is 5 different supplements, NDGA, resveratrol, urolithin, glycine, caffeine. And I think this panel represents really a good sample of what are the drugs that people are considering that extend lifespan in worms, but even in also other model organisms potentially and in humans. So the setup that we use to test this drug, I'm going to go relatively quickly on this, but we do it in a more automated way that most labs do. We put worms in 24 well plates with agar, and we take pictures of them every other day. And if from one day to the next we see that the worms didn't move, then we consider them dead. And I'm going to show you a video of this. So on the left part, you have a control well with worms. This part is the bacteria. And then you have a treated well on the right part. And also what we implemented is a machine learning algorithm to detect the dead worms. So we do that to detect when the worms die. So we do that automatically, so we don't have to do it ourselves. So I'm going to just play the video, and you will see as the worms die, there's a red square, which means that they are being picked up by the algorithm as automatically as dead. And if you pay attention on the treated well, it's going to take a bit longer for the worms to die than on the control well. All right, so this is our system. This is how we test drugs in worms, which, as I said, is a bit more automated than what most labs do, although there are some other labs that also try to automate these testings. So now I'm moving on to the results. So first I'm going to show you the results of the drugs when the worms are fed with live OP50. So these are the 16 drugs that I talked about. So you see that a lot of them do work quite well. So for example, resveratrol, Rapamycin, urolithin, metformin, GSK, LY, CCP, caffeine, captopril, they work quite well. But there are some drugs that do not work at all. Aspirin, glycine, NDGA, simvastatin. So also these drugs, we tested them at least at 3 concentration, oftentimes at even 5. And we show here the result of the best concentration. So for example, if I say here that glycine didn't work or NDGA didn't work, it means it didn't work at any of the 5 concentrations that we tested. Now I'm going to move on to the result with the UV-killed OP50. But before that, something which is important to know, and it's been published before, is that in general, worms live longer on a UV-killed diet. So you see just control worms with the normal diet and the UV-killed diet, and they live quite longer. So now moving on to the result with the UV-killed bacteria. So some of the drugs still work. Doxycycline, GSK, simvastatin, resveratrol, LY, they work still well with the UV-killed bacteria. But there are some drugs that used to work well with the live bacteria, but don't work anymore. So Rapamycin, for example, urolithin are examples of those drugs. And I'm going to show you a recap in the next slide just now. So here I'm showing side by side the effect of the 16 drugs, both in the live condition on the left and in the UV-killed condition on the right. So we found that the drugs can be separated into 4 different groups. So the first group is the drugs that didn't work at all, either in live or UV-killed. So you see aspirin, glycine, rapamycin. So what could be the reason for that? I'm going to talk a bit more at the end, but in general, we think either it's not reproducible or we missed the right concentration, although we try to test close to what's in the literature. But as a matter of fact, for us, these drugs didn't work. Now, the second group is the drugs that work well both with live and UV-killed OP50. It doesn't mean that they have exactly the same effect. So you see that, for example, GSK works better in UV-killed than in live, and resveratrol work better in live than UV killed, but generally they work in both conditions. Then there's quite a few drugs that only work in the live bacteria, and I'm going to tell you our hypothesis for that. And lastly, the most surprising group is there are a couple of drugs that only work in the UV-killed bacteria. All right, so as I showed you, most of the, and it's quite good in a way, it's quite reassuring. Most of the 16 drugs that we tested, which are supposed to be the drugs that are best reported to extend lifespan of worms, most of them have some effect on worm lifespan. And if I single out some of them, resveratrol worked amazingly well, and it's interesting because it's a bit of a controversial drug, but at least in worms with the live, especially with the live OP50, it works amazingly well. It was one of our best drug. Metformin worked well. Also, I want to mention that both of these need to have a quite high concentration to be able to work with worms. And it doesn't mean that in human we will be able, for example, to reach the same concentration. I know in human with the resveratrol there's some issues with the bioavailability, but with metformin, it's also for human, it's given at quite high dose. Rapamycin didn't work, so that may be surprising because rapamycin is one of the best drugs for prolonging lifespan, especially in mice with some of the most robust results. But at least for us in worms, it didn't work, and we're not the only group to report this. So the CITP in the US, which is also a consortium testing drugs in worms, for them, rapamycin did not work, and another company in the US called Aura, for them, rapamycin didn't work at all. So maybe it's not stable in the agar, that's one hypothesis, or maybe it's just precipitating out of the agar, which is also something we saw that could explain this. But on the other hand, other mTOR and PI3 kinase work quite well, so that's reassuring. And as I said, the effect of the drug is largely mediated by the bacteria, meaning we see differences in live and UV-killed food. And for that, we have some explanations. I'm not going to go into too much detail because I want to talk about other things, but we think either sometimes the drug is toxic to the bacteria, and this is what extends lifespan of worms, because we know that the worms live longer with the with a killed bacteria. Or in other cases, maybe the drug is metabolized by the bacteria, and this is what explains its effect. But there's a paper that recently came out, and I invite you to take a look if you want more details. So that's it for the worm part. And this is the organism that I'm going to talk about the most, so the rest will be relatively shorter. So here, this is in flies, we have built a similar platform. So we also built a platform in flies to test drugs for lifespan extension in a relatively automated way. I'm going to just show you this platform. So it's the same principle. You have the flies go through their lifespan with the drug in the food, and as they die, they get picked up by the machine learning algorithm. And you see here, again, in the control group, the flies die faster than in the treated group. Yes. All right, and then a few results, this time very briefly, still on some of the drugs I was showing you before. So we saw that doxycycline, so it's tested this time in two genders, female and male. The worms are just hermaphrodites. So doxycycline worked well in flies, metformin also working well, rifampicin also working well. Some drugs not working as well in flies. So that's all for the flies. Now I'm going to move on to the fish and mice. So we also have this platform to test drugs for the whole lifespan extension in fish and in mice. The difference, I would say, is with these models, we didn't try to improve that much the automation of the process because, I mean, it's still pretty manual. But what we try to do is to decrease the cost of the experiments as much as possible. So for that, we are working with some animal facilities in the south of Europe, in Spain, and we're able to run lifespan experiments with fish and with mice in a with a very decreased cost. And for example, we have the numbers that are announced by the ITP in the US or by the Lev Foundation, and we estimate that we are 9 to 15 times cheaper to test drugs for lifespan in mice, for example. And I'm not going to show you results today because we're still analyzing them, but we can do this at a very cheap cost. And lastly, for the mice, something we also do is not only test if the drugs can extend lifespan, but also if the mice have a preserved functional ability with age. And the way we do this is by using the digital ventilated cages, which track activity for 24 hours every day. And you see an example here, which is 2 weeks and a half. So you have the day. During the day, they are less active, and during the night, they are more active. And you can track their activity for 24 hours throughout here 3 weeks, but throughout the whole lifespan. And when you test the drug, you can tell if the drugs that extend lifespan also are able to preserve activity in the mice. All right, so as a conclusion for this part, we have established a screening platform for lifespan extending drugs in 4 different models: worms, flies, fish, and mice. To answer the conservation question is still too early, but there are some signs that some drugs are conserved between different models. For example, I showed you between worms and flies. But the issue still remains that, especially in mice, it's very lengthy to test these drugs. We only give them at the last part of the lifespan, which is the last year, but it's still quite lengthy and costly. And people are trying to develop biomarkers of aging or predictors of mortality to see if we could get this answer faster, but it's still to be validated. All right, and now this is the last part of the talk, and I'm almost reaching the end. So humans. So we are also interested to see if there are drugs that extend lifespan of humans, and if it's already possible to know that from retrospective study. So we're not the first people to have this idea. Initially, the idea came from metformin, which is a drug that was shown. It was shown that people with type 2 diabetes taking metformin were living longer than people not taking metformin, which was quite surprising because you could think these people have diabetes, they should live shorter. But no, from a retrospective study, they were seeing that they lived longer than people not taking the drugs. And our idea was, okay, this is interesting, but why don't we do this with all the drugs that people are taking and see if some of them are associated with longer lifespan? Maybe that's possible. So the way we do this is we went to the UK Biobank, which is a very large biobank with almost 500,000 people, 12 years of follow-up, so it's pretty long follow-up. And more importantly, they recorded around 56 million prescription medications. So we have a lot of medication data. And we did the same that's in the metformin paper. So we compared people taking the drug with people not taking the drug and compared their average life expectancy. The problem is that people taking the drug often have confounding factors. So you cannot do it just like apples to apples. You have to do something, and that's basically called matching, which is very common in epidemiology. So what you do is you compare people taking drug A with people not taking the drug, but you restrict this sample to the people that have exactly the same confounding criteria as the people taking the drug. So in this case, same proportion of diabetes, same proportion of smoking or male gender in both groups. And then you can compare relatively safely the different drugs. No, sorry, the people taking the lifespan of the people taking the drug and the people not taking the drug. So what are the results? So we were able to analyze 406 drugs that were taken by at least 500 people. That was the threshold we set for sample size. And 169 of them had a significant association with survival after correcting for multiple comparison. And this is how the hazard ratio looks like. So more than one is decreased survival, less than one is increased survival. So you see that most of the drugs have associated with lower lifespan, which is normal because usually the people taking those drugs, they take it for a reason, a disease that may reduce their lifespan. But we still found 14 drugs that were associated with a longer lifespan. So what are these? And this one was morphine, by the way, which is, for obvious reason, associated with shorter lifespan. So what are the 14 drugs that are associated with longer lifespan? One of them was a statin, atorvastatin. There was Viagra, sildenafil in male, where male live longer. And then very strikingly, there were a lot of estrogen. So estroderm, vagifen, estriol, estradiol, all of them containing estrogen. The women taking these drugs were living on average much longer. And the hazard ratio was quite significantly protective. I'm going to skip this because I'm out of time, but there were effects in both genders. So we saw effects in both male and female. We also saw dose response effect because the biobank also records which dose, the dose at which the people are taking the drug. And we saw some nice J curves for the dose response on the protective effect on lifespan. And then lastly, we tested the effect of specific drug class on lifespan. So we pulled the drugs together and we looked at estrogens, we looked at PDE5, which is the class of Viagra, SGLT2, statins, and metformin. And here, so the estrogen, again, were working very well, the PDE5 also. SGLT2 was very strong, although the sample size was quite low, so it was just around 500, which is why the curve looks like this, but it was still having a strong effect. And the rest was a bit less significant. And metformin, which I talked about in the introduction as the introduction for this work, the idea here, it did not work for us. It doesn't extend lifespan. So very briefly, to discuss this, so we have identified 14 drugs associated with lower mortality in the UK population. Some of these drugs have very interesting literature. I'm not going to enter into too much details, but there's a lot of literature on estrogen and hormone replacement therapy and how it may or may not be beneficial for menopausal women. Statin, obviously, a lot of literature. Viagra was found in another study also to prevent Alzheimer's disease, was also a retrospective study, but using a different biobank. Metformin did not work, and we're not the first one. So there was already a paper in 2021 that reanalyzed, I think, their data or also used data from another study and showed that, in fact, metformin was not associated with increased lifespan. Obviously, we cannot account for all potential confounders, only those that we have. And this is why some of these drugs may increase lifespan for other reasons than the drugs themselves. And then the last question is, should you start taking these drugs? No, I think these are retrospective studies, and you should wait for you should wait for clinical trials. Now, how to design these randomized clinical trial is another question, but I'm not going to enter into this today. Or maybe during the questions. All right, so this also study was published quite recently, received some attention in the press because people are also asking if they should be taking Viagra or statin or HRTs. But for me, again, no, it's too soon. And then, yes, to conclude, I thank everyone in the team, and I also thank the audience, all the people attending today, for listening. Thank you.
Thanks, Kevin. Lots of questions. You mentioned something at the beginning that there's lots of, and maybe I didn't hear you correctly, that there's lots of positives reported. And of course, people don't report their negative data. Our lab's been somewhat guilty of that too. We have a lot of things that don't work in mice we haven't reported. But on the other hand, if you look at the screens in worms or yeast where you can do genome-wide screens, there's a surprisingly large number, at least, of genetics mutants that extend lifespan, and probably drugs too. So I've come to the conclusion that it's, at least in these non-vertebrate models, it's probably pretty easy to extend lifespan. And I agree with you. There's the reproducibility issue, which we can come to in a second. But would you agree with that?
Yeah, I completely agree. So, I mean, in general, the worms, especially in flies, it's a bit more difficult. In yeast also,
Yeah.
it's quite easy. So the worms are quite sensitive to those intervention, and there are a lot that seem to extend the lifespans. I think it's also important to look at confounding factors. So, for example, any antibiotic or any drug that can kill bacteria, if you test them with the live bacteria, it will lead to an extension of lifespan. So that could be a big one. And otherwise, you also have to look at the threshold. So a lot of drugs extend lifespan a bit. It might be significant. But I think if you start setting like stronger thresholds, like 20% extension, 30% extension, and with the UV killed bacteria, then you will have a lot less drugs that.
If you put that criteria in flies, though, you wouldn't get anything. I think 20 to 30% is a huge effect for flies. Why is it so difficult in flies?
So we've worked a bit in flies. I showed this automated testing also. It's definitely much more difficult to manipulate them in extended lifespan than worms. I don't know exactly why. I would say that in general, the longer you live, the more difficult it is to manipulate to extend your lifespan. So I would say that then in mice it's more difficult than in flies, and probably in human is going to be even more difficult. But otherwise I don't know exactly why.
Yeah, I've been puzzling with that either. Just a note on rapamycin and the worms. We have a problem in yeast replicative lifespan. We could never get it to extend, and the reason was that when you put it in the agar, it's just not stable long enough, and that's a reasonably long assay to do in the yeast. And I think that we kind of see the same thing in worms. If you're doing it on an automated system where you're leaving the rapamycin on a plate, as opposed to switching plates, we don't get much of an effect. So given that the other mTOR inhibitors still work, my guess is that's what the problem with rapamycin is.
Yeah, I completely agree. So as you're saying, so the way we do the lifespan in worms, we don't transfer the worms at all. And so they have to stay with the drug for their whole lifespan. And I mean, stability of rapamycin is well known. In mice, when you give rapamycin to the mouse, you have to encapsulate it because otherwise it gets degraded. I mean, it's not really comparable, but in general, I think that might be the issue. And the fact that other mTOR PI3 kinase work, I think confirms that still this pathway is one of the most important.
I think it's really interesting with the urolithin because we see very strong effects on urolithin in mice too, so we're quite interested in that molecule. The urolithin in mice or humans has to be metabolized, right, in the gut. It's really a precursor for active ingredients, and so I'm wondering what the bacteria are doing to urolithin. Are they creating a metabolite of it that's active or more active potentially? And can you look at that? I think that would be a fascinating question.
Yeah, exactly. I mean, we really wanted to do that because of that, because urolithin is known that in the human gut already it's metabolized, and we saw in worms that it was working better with the live food than the UV kill. So we thought, yeah, it's probably being metabolized, and we wanted to do some mass spec to try to identify if it's still there or not and what is potentially the new compounds, but we haven't done any of that. I mean, doing mass spec would be also a follow-up for other drugs to see why or if they are being metabolized. I don't know if there are other ways to do this, but this is the way we thought to do.
Yeah, I think that, we haven't done much of that. We've done some tracing inside mice, but we haven't looked directly at the bacterial metabolism. But I think that this is kind of a missed area in the worm field, and somewhat in the fly field too, because you can use different strains of bacteria or yeast for food, in theory. You can also use genetic mutants of bacteria and yeast for food sources for worms and flies, respectively. And that might be a really interesting way to look at the interaction between drugs, bacterial mutants, and worm mutants in the context of aging. I know it gets to be a big problem as you start modifying different things at the same time, but I always thought there was something really interesting to learn from that.
Yeah, no, for sure. I mean, in worms, there is a strain of bacteria called HT 115 or something like that. Maybe not exactly, but the worms also live longer in this particular strain of E. coli. And we thought to also test with this. Other people have asked us also to test with it. Or what some people ask us is, can we just test without bacteria? Because, the worms can also live in, it's called axenic medium.
Yeah.
But it's a bit difficult to handle. It's not very reliable, so we didn't do that. But yeah, I think that there are a lot of ways to try to continue to explore this part. And yeah,
I don't think it's very easy to automate either, because in azenic media they're in a swimming condition, aren't they? Or typically,
yeah, that's also the issue.
yeah. But it's an interesting question. You mentioned that it's much easier to do. I want to get to the human stuff in a minute. You mentioned it's much easier to do or cheaper to do the mouse studies at the laboratories in Spain, which is, I think, exciting. Um, is it cheaper at the same scale the ITP is doing, or what kind of numbers and power can you do in the mice and one other thing I think is one thing ITPI doesn't do much of is to really do a lot of health span measures. It would be great to look at other properties of the mice, particularly if it's cheaper to do the experiments.
Mm-hmm. Yes, so yeah, for the scale, yeah, definitely. So we were testing even, I think, more drugs per year than the ITP. But so they use three different centers, right? And each center has 50 mice, and we use just one center with 50 mice. So we kept like the significance, but as one center, and ITP has three, so it's difficult to compare. But yeah, when I say it's cheaper, I'm normalizing for the number of mice, etc. For sure. And what was your other question? Sorry for the.
Can you look at other health span parameters during the aging experiment?
Yeah, so this we thought a bit about this because sometimes the health span is even more costly to do, or more work to do than the lifespan. And that's the nice thing with these DVC racks, is that they are recording. You don't have to do anything. You get the data, and you get the activity and live without having to do anything. And we were quite surprised because, and these results we didn't publish yet, but regardless of the effect on aging, a lot of the drugs we put, they completely shift the patterns of activity of the mice. And that's quite...
that was my next question.
That's very striking. Yeah.
That's fascinating. And also in the context of circadian rhythms, I wonder if you can ask whether the mice have whether they're shifted or not shifted, whether they're more normalized in terms of their activity. Mice lose that somewhat lose that distinction between day-night activity as they age. I'm wondering if it's preserved in the drug context.
Yeah, exactly. I mean, we see this. We see that the activity is less clear, between the day and the night. Also, there's a very important factor in aging, but I don't think we are the first to see this, is the napping. So even at night, their pattern activity is not continuous, even in normal condition. They nap. And these patterns are really changed with aging, and also with the drugs is quite striking. Some drugs make sense. I mean, we tested some drugs that, I don't know, are supposed to affect maybe sleep, and these you see it, so it would make sense. But other drugs we didn't expect, and still it changed quite a lot the activity.
Interesting. Just quickly on the humans before we go to questions. I think that first of all, I'm directing a reproductive longevity center that focuses on ovarian aging, and so we've looked a lot more at this hormone replacement question. And I've sort of come to the opinion that the question shouldn't be should a woman go on hormone replacement uh, during menopause. It should be, is there any reason they shouldn't go on hormone replacement? Because the data is so positive for most women that aren't at high risk of various other um, factors that might be exacerbated by the hormones. But for the most women, it seems like that there's clear benefits. And that seems to be what your data is saying. I mean, I know you don't want to encourage people to start taking drugs, but women are already making this decision on hormone replacement. So what are your thoughts on that?
Yeah, no, I completely agree. That's what the data seems to show. I was also contacted by some doctors that already prescribe these to their patient, and they were telling me, yeah, it's great because now you also see this in your data. And so I think there's a lot of interest for this. There's another thing to know is even in mice, the ITP showed that alpha estradiol extend lifespan of mice, but in males. So that's another way. But yeah.
Just quickly, speaking of males, let's talk about Viagra. It's not that surprising that it would extend lifespan. It's improving blood flow, and that was likely to have improvements in preventing a range of complications of chronic diseases. So I don't find it that shocking that it would extend lifespan. It may actually be a decent longevity drug, I think, at the end of the day.
I agree. So that could be one of the mechanism. Of course, is the drug that I would say people complain a lot about the confounding factors, because it's true that there could be many. Also, PDE5 might be an interesting pathway for aging, because even in worms where it has nothing to do with blood flow, in the paper, I think it's in the paper, we show that tadalafil extend the lifespan of worms. So maybe there's also some conserve mechanisms there.
Yeah, it's increasingly a wonder drug, and it was almost abandoned. It's amazing.
Yeah.
Yeah, it's fascinating. It's also
already
very safe,
repurposed.
I think.
It's already a repurposed drug,
Yeah.
right? But it might be
And also
again
very safe. I mean, given as many men that take this off-label without medical supervision as there are,
Yeah.
I don't recommend that, by the way, but lots of men do that. It's remarkable how little side effects that emerge from it. So it's an interesting drug. I want to bring in Li Huan Guan to ask some questions from the audience. Li Huan?
Thank you, Brian. And thank you, Dr. Perez, for your wonderful talk. We have many questions from the audience. I'll start with questions on the animal results. So are the bacteria identified? If yes, then are they present in a pathologic human microbiome?
Yeah, I mean, we know the type of bacteria that we're giving to the worms is a strain of E. coli, but I don't know if I mean, of course, in the human microbiome, there is E. coli. I don't know if we can exactly compare, but it's true that in general, the fact that some of the drugs we take as human, their effect might be mediated by the microbiome. This has been shown, I think, even in the field of cancer. some anti-cancer drug, it has been shown. And yeah, we could make the parallel. Mice have a microbiome, humans have a microbiome, and the effect of the drugs on lifespan could also be mediated by this for sure.
And certainly, some of the drugs do change the microbiome. That's already known with rapamycin and metformin. There's no causality to that.
Yeah.
There's no causality to that for aging yet, but it's certainly feasible that that could be the case.
Okay, thanks. The second question is, have you considered assessing life's extension effect of combination therapy? For example, 2 or more drugs together and instead of just monotherapies. Might be interesting to see synergistic and antagonistic effects.
Yeah, this is very important. So in the worm paper, we show some very strong combinations. We show, for example, GSK plus doxycycline having a very, very strong effect. And I think that, as we said at some point, that in mice or human, it might be more difficult to extend lifespan than in worms. I think we're going to need combinations. And worms are a great model to test combination because you could do combinatorial experiments. But yes, I think this is very important. But it's not very often that you would see, I mean, from what we've tested, it's not that often that you would see synergistic effects. So sometimes people expect that if you just take 2 drugs that extend lifespan, the effect will be additive or synergistic, and most of the time it's not the case. So you really have to test them. Sometimes even can become detrimental. So
Yeah. We see the same thing.
In other animal models than worms, do you think there will be other external factors such as the OP50 in C. elegans that would lead to non-lifespan related extensions?
Yes. So in flies, there is a bit the same issue because the flies, if you just maintain them the normal way, there is still a layer of bacteria that develops. I mean, they're also not sterile. So we thought maybe antibiotics, because again, doxycycline works in flies, could be also having the same effect on the bacteria. We didn't test this specifically. So in worms, I didn't show it during the talk, but we had a very specific assay on this where we tested the effect of the drugs directly on the bacteria without any worms. And we saw that sometimes the drugs was toxic to the bacteria. For the flies, we didn't do this systematically, but we thought potentially that might be the case. Otherwise, yes, there are a lot of confounders for lifespan. I mean, for worms and flies, there is the temperature, which is an obvious one. So if you decrease temperature, they live much longer. Yeah, the food, because of potential calorie restriction, could be an indirect effect. So yeah, there's a lot of confounding factors.
Yeah, and that's certainly true. It gets even worse as you get into mice and certainly humans. I mean, I'm not talking about the cinnamon roll I had this morning. I needed sugar to wake up.
Another question is, were there any drugs that surprisingly correlated with decreased lifespan, and how might this shape future safety discussions?
Yeah, that's an interesting question. So I think worms have been proposed as a model to do toxicity screening, and I think it would work really well. And we also have this idea to do some just toxicity screening. Now, is the effect going to be conserved? Is the same question. If the same way we don't know if the lifespan extending effect will be conserved, we don't know if the lifespan shortening effect will be conserved. But in general, if you're going to develop a drug for humans or even testing it in mice, if you know beforehand that, okay, you might do toxicity assays in cells already, and you can see that it's toxic to the cells. But if you know in advance that it's toxic to the worms, that may be one less reason to test it in mice.
Okay, so now we move to the questions about human. Is the analysis on UK Biobank matched by socio-economic status? Because some of the drugs are expensive and unlikely to be covered by insurance. People who are taking it may have a better financial situation.
Yeah, it's a good question. So there is one way we do that in the UK Biobank is with it's called the Townsend Deprivation Index. We match for that, and it's related to the socioeconomic status. And I think for one of the drugs, I think for estrogen, we saw that people with a higher Townsend index were more likely to take estrogens, but we completely account for this. But of course, I don't know if this index, I'm not really an expert on that, but I don't know if this index would account for all type of socioeconomic status.
Yeah, the matching is challenging too, in these things. We've been doing this within Haynes, and one of the things we noticed in this collaboration with Jan Gruber and Feng Sheng, one of the things we noticed is that there's a 20% of the people in Haynes. So this is around 2000 in US. 20% of the people around 60 to 65 have some medical condition, because we have their clinical markers, but they're not taking drugs for that. So they're basically untreated. So when you do matching, you're either matching to people, if you're using a drug to treat a condition, you're matching to people that have that condition or taking a different drug, or matching that condition that are not being medicated. And that second part is challenging when you're trying to do survival, I think, at least.
Next question is, how likely the results from the animal models will translate to humans?
Yeah, I wish I knew. As I said, I mean, for now, it's a bit early to answer this question. I think it's first easier to answer it with the animal models, so to see if drugs that work in worms will work in flies, if drugs that work in flies will work in mice. You can start to build those tables, but still we need probably more data than we have currently for this. For humans, I think, yeah, from mice to human is the same question for any biomedical research field, right? You test drugs in mice, you don't know if they're going to work in human, but for now it's the best we can do.
Okay, we'll see. I think my last question is a technical question. So does the resolution of photos influence the algorithm to identify dead animals, organisms?
I guess if you mean that you want to reuse our system, which is currently not public, we might publish it at some point, but the machine learning detection is not published. But in that case, we tested it with a few different setups, but I don't think I could answer exactly. But I hope that other people would be able to reuse it, or that they would need to just get the same setup that we have, which is not too bad, because in the end it's just one microscope. That's something we prefer to the Aura approach, where you have to get several robots, several worm bots, and we only need one microscope. And just I remembered for your previous question, I think even if we don't necessarily know yet the answer for the conservation about the drugs, we could still look at some genes. And if we see that, for example, the mTOR pathway or PI3 kinase pathway, they are conserved in multiple, in mice and in worms, or in flies, you can already get some idea of conservation. And the other example that people often give is with caloric restriction. That has, I know that there are some caveats to that, but still it has been shown that in multiple species, creating a calorie deficit would extend lifespan. So that's also one way to answer this.
Great. Thanks, Li Huan. And thanks, Kevin. That was really interesting. We'll look forward to more data in the near future, hopefully. And with that, I want to remind people to use the chat function, and panelists and all attendees option to leave comments and feedback. We have a program promotion. The Healthy Longevity Intensive Course 2025 will be from June 30th to July 11th at NUS. and applications for that are open. That's 10 days intense interaction, cutting-edge learning, and unforgettable experiences on geroscience and healthy longevity. The next Healthy Longevity will be with Jorming Go, one of our very own, a research assistant professor here at NUS. He hasn't given us a title, but it will be something to do with exercise physiology and aging, I'm going to guess. And with that, I'll leave you with our final video. And to end today's session, it's a short video highlighting how music can be a powerful tool for healing and connection. It's a reminder that creativity could be a vital key in health and well-being. Thanks for joining us, and we'll see you next time.
What a wonderful world. Music functions as something that brings us all together. I don't think I've ever met anyone who doesn't like music or has some sort of connection or relationship to music. There's such an important healing value to expressing yourself. The dark, sacred night. Is what is life without being able to express yourself in a beautiful, meaningful way? What a wonderful world. While a counselor, social worker might use a modality of talk therapy, or a recreation therapist might use games or recreation, music therapists are trained to use music. We are uniquely trained in all the different ways that music can help and heal and be an alternative treatment. I spend part of my time on inpatient adult psych, and the other half of my week is spent doing medical consults and medical music therapy. Each patient is different, and so the music therapy might look different. A patient who is struggling to find a sense of meaning and connection because of their mental illness, and their illness is a barrier to that. Oftentimes, what I hear from patients is music is an expressive outlet. A lot of times patients will come into group, they're not sure about it, but then you give them a drum and they're just like, what is this for? And then you lead the group session and you see the smile on their face. Or if a patient's having a really bad day, if I can sort of break that tension and act goofy and sort of pull them out of it. You know, music therapists are trained to utilize that in a therapeutic way. Very soothing. They talk about songs that are meaningful for them and the ways that music is an expressive outlet for them. It's also such a great feeling to come out of a group session and the patients were like, oh my gosh, this is the most fun I've ever had in a hospital, you know? I can honestly say that I'm so grateful to be able to be in this really unique role.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.